Okay, good morning, everybody. Thank you for coming. So welcome to the Light Sheet Imaging at Universidad Mayor, or Lyceum, series of webinars. We're supported by the Chan Zuckerberg Initiative to expand global access to bioimaging. And our main objective is to build a light sheet imaging hub in Latin America. So today, we're co-hosting this webinar with LibreHub, also supported by CZI, and whose mission is to expand access to open hardware instrumentation for biomedical researchers in Latin America. So through Lyceum, we would really like to network light sheet researchers and share and troubleshoot approaches using this technique to answer biomedical research questions. So you can visit our website at lightsheetchile.cl to learn about new events and to join the Light Sheet network of researchers. And I will also post the recording of the seminar there. In addition to organizing these webinars and workshops, we're very lucky to be collaborating with today's speaker, Dr. Jan Huisken, to learn how to build this small and more affordable light sheet microscope called the Flamingo. So with the Flamingo, we really hope to spread more light sheet microscopy in South America. And Pamela Mina, a postdoctoral researcher who's been working with Dr. Huisken's group and traveling to Germany to learn the process, will now be introducing Dr. Huisken.
Hi, thank you, Alenka. Yes, I'm super happy of introducing Jan and his talk. So, but before introducing him, I would like to welcome you all to this two-part webinar. In the first part, Jan will describe shortly about light sheet fluorescence microscopy development and the applications in his lab. This talk will last approximately 50 minutes with some time for a couple of questions after that. And then we will have a short break. And at 11, we will start with the second part, which is a demo of the Flamingo system. And there we will have questions and answers about this, this microscope. Voy a aprovechar de cambiar un poco, darle la bienvenida a toda la comunidad hispanohablante y quiero decirles que lo que vamos a hacer es que todas las preguntas que ustedes tengan las pueden hacer directamente al chat, escribiendo directamente a Alenka o a mí, y nosotros haremos estas preguntas al final de la sesión. Estas preguntas las pueden hacer tanto en español como en inglés. I will switch to English again. So for the questions, if you have questions, you can write the questions directly to the chat, to Alenka or to me. And at the end of the talk, we will provide the questions to Jan. Okay, that being said, I would like to shortly describe Jan's trajectory in science. So he's an Alexander von Humboldt professor at the Georg August University in Göttingen, in Germany. He studied physics in Göttingen and Heidelberg, and he has a background in three-dimensional fluorescence microscopy, optical manipulation and trapping, developmental biology, and zebrafish development. He received his PhD from the EMBL Heidelberg, where he pioneered multidimensional light sheet microscopy, also known as selective plane illumination microscopy, SPIM, in the labs of Ernst Stelzer and Joachim Wittbrod. For one of the first applications of light sheet microscopy, Huisken moved to the lab of Didi Steinier at the University of California, San Francisco as a cross-disciplinary HFSP postdoctoral fellow in 2005 to study cardiovascular morphogenesis and function in seabirds. From 2010 until 2016, Huisken was an independent group leader at the Max Planck Institute for Molecular Cell Biology in Genetics in Dresden, Germany. From 2016 until 2021, he was a principal investigator and director of medical engineering at Morgridge Institute for Research and professor at the University of Wisconsin-Madison. Jan is now best known for his interdisciplinary work at the interface of general high-resolution microscopy and quantitative developmental biology. His lab covers all aspects of modern in vivo imaging, from sample preparation to image analysis. Recently, he has started an initiative to democratize the access to advanced microscopy with a modular and portable microscope platform called Flamingo. For his contributions to modern optical microscopy, Huisken was awarded the Royal Microscopy Societal Medal for Light Microscopy in 2017, the Lennart Nielsen Award in 2020, and the Humboldt Professorship in 2021. So with this introduction, I'm super happy and excited to hear what Jan will talk about. Thank you.
Okay, thank you very much for the nice introduction, Pamela. Thanks everyone for joining. So today's talk is going to be on light sheet microscopy, in particular the Flamingo, which we already see here on the first slide, and a lot about how to image zebrafish with light sheet microscopy. I must say light sheet microscopy now is a very wide field. And what I'm presenting here today is just a really narrow view on things that are interesting for us. This is not an attempt to give a general overview of all the things that are being done in light sheet microscopy. Would probably also take much longer than just an hour. So as Pamela said in the first part, I'm just showing you some slides. Showing what we've been doing in the past years and where I think light sheet is heading, at least in the area that we are concerned about. And then the second part, I will be in front of a Flamingo. I wouldn't necessarily call it a demo. We'll see what we can do, but it's mostly about showing you the system so you see what it looks like, and I primarily want to give you an opportunity to ask questions. Where is this? Where is that? And then I can point at it and show it to you live at the microscope. And of course, I'll have a sample as well, and I can show you how the imaging is being done. Good. So let's start with this part. I've split this talk into 3 parts. The first will be sort of giving you a general overview of some of the main projects that we've been dealing with. And the focus here is really not to give, to show you everything and to dive into the science of everything, but more to give you a sense of what we've been focusing on in the lab and what our view on light sheet microscopy is. And I think a particular emphasis will be on the customization of light sheet microscopy for specific applications. And then this will also be a focus for the second part, where I will sort of discuss briefly where we see issues with how microscopy development is being done in optics labs and how we could potentially make this more sustainable. And then the third part will go into Flamingo, which is not going to be super long because we'll have the second part directly in front of the microscope. So for the first part, I want to start by emphasizing how multidisciplinary everything is that we've been doing and still doing, and emphasize that it's not just a bunch of physicists that try to develop cool microscopes, but it's a lot about the interplay between all these different areas that I've highlighted here, in particular, of course, the biologists and their questions and their problems that we try to address with custom-built high-end microscopy. But of course, there's also many other people involved. The image analysis, I think, is an obvious topic as well, especially in light sheet. But this, what we call here the imaging scientist, I think is an essential component, someone who sort of tries to communicate between these different areas and tries to especially translate between the biologists and the computer scientists, and something that CZI has been pushing a lot, and we're really thankful for their initiative to fund numerous imaging scientists around the world. So light sheet microscopy, as I said, has now taken lots of different shapes and has seen lots of different applications. This probably summarizes very well how we do light sheet microscopy. And at the same time, I think it summarizes quite well sort of the main fundamental components of a light sheet microscope. In this case, we have two distinct optical paths, one for illumination, one for detection. These could actually be done through the same objective lens, which some people do, but in most cases, it's a lot easier using two distinct optical paths. So we have an illumination objective lens that projects a laser light sheet into a chamber, typically onto the specimen. And then perpendicular to this illumination axis, we have a detection axis, which collects the fluorescence that is excited in this one plane that is being illuminated by the light sheet. The other thing that's indicated here is that the tube in which the sample is embedded doesn't need to be a tube necessarily, but in this case it is, can be moved in all dimensions, and it can also be rotated, and that adds a lot of benefits, particularly when we're trying to image an object that's larger than the penetration depth of the light sheet or of the detection system. The detection system is not shown here. It's very rudimentary. It's basically an objective lens, a filter wheel, a tube lens, and a camera. We can look at that on the Flamingo in more detail. So this is what it typically looks like. So here we see two illumination lenses. That's already a hint that this system can be expanded in various ways. So here we have three objective lenses, two for illumination and one for detection. And the sample would be placed directly in the middle, where you see the little light sheet there between the two illumination lenses. So to jump ahead and show you what a typical light sheet data set may look like, this is a maximum intensity projection of a 3D time lapse. And we're looking at neuronal development in a zebrafish, where like a special family of neurons here is labeled. And we also see the lateral line now developing in this fish. This is a time lapse over like 18, 20 hours or so. And what I want to highlight here particularly is that the challenge is not so much in the actual microscope, but it's more in the sample preparation. And the reason why this video has already won some awards was really that we've been able to immobilize the fish and orient it in exactly the right direction to take this data. And of course, still keeping the fish happy during the entire duration of the time lapse. And oftentimes that is really the key to success with light sheet microscopy nowadays. It's less about the specifics of the microscope. It's more about how you build the microscope around the specimen and how you prepare the specimen. So one slide I cannot skip is really sort of this comparison between point illumination and detection, like we do in a confocal microscope, and light sheet microscopy, which is shown on the bottom. And it's worthwhile emphasizing that every time in a confocal, oops, sorry. Sorry, I have to, there's an update that just happened. Okay, are we back? Okay, so it's worth noting that every time you're taking a single plane in a confocal microscope, that the sample is always illuminated. The entire specimen is illuminated. And in contrast, in a light sheet microscope, when you take one image, only this one plane ideally is illuminated. And so that, of course, reduces the phototoxicity dramatically and makes it a lot more efficient in how the fluorescence is used and collected on the camera. So that's shown on the left. On the right, we have the difference between the point detection in the confocal and the full frame wide field detection in the light sheet. And it's important to understand that moving from a point detection in the confocal, where you only have microseconds to collect the fluorescence per pixel, to the light sheet mode, or the light sheet detection, where the entire chip is read out at once, you have milliseconds of exposure time and milliseconds to collect the fluorescence. So even if you're running the camera at, let's say, 100 frames per second, you still have 10 milliseconds of exposure time, which is orders of magnitude higher and longer than what you have in the confocal. And that makes the light sheet give you these beautiful images, the high dynamic range, the low noise, and really allows you to also process the data very nicely. And of course, you can push it if you want to high speed. We've taken videos at like 5,000, 10,000 frames per second, in which case you're using more laser power, of course, and you're shortening the exposure time, but it's still within range and it's still within what the sample tolerates. So as I mentioned, light sheet microscopy has gone in all sorts of different directions, and the main directions that I see are sort of summarized here in this slide. The focus that we've had always right from the beginning when we started with light sheet was on the organismal level, so dealing with sample sizes around a few hundred microns up to a few millimeters. And more recently, we've seen a push also to use light sheet microscopy for clear tissues, which are more in the millimeter to centimeter range. And interestingly, light sheet microscopy is still the tool of choice for imaging these specimens, because it's again the speed and also a little bit the low phototoxicity that make light sheet ideally suited for imaging these large specimens. And then on the other side, we've seen light sheet also being used for much smaller scales, individual cells, which typically runs under the name of lattice light sheet, but in fact, it's something you could do with a Gaussian light sheet pretty much in a similar way. It's just that was sort of coined as the term for when light sheet is being used for such small specimens. And in fact, one can show, but that's another topic, that most of these lattice light sheet experiments could have been done easily also with a conventional Gaussian light sheet. So what I want to emphasize with this is that light sheet is really pretty universal, and it's just a matter of how you shape the beam, how you, what kind of lenses you're using, what your desired field of view is, and what your specimen is, how your specimen is most happy, and then, as we like to say, how we build the microscope around the specimen. So a lot of what's needed for getting the perfect experiment is really tailoring the microscope to the application. And that's essentially what we've been doing for the last 10, 15 years or so. Okay, so just to give you a few snapshots, sort of to emphasize also the multi-scale aspect. Multi-scale means that you're trying to image different processes at different scales and maybe different specimens, but oftentimes it means that you're trying to look at something that is happening across multiple scales in the one and the very same specimen. So here in this case, we are looking at the first few cell divisions in a zebrafish, which is happening more on the scale of a few hundred microns. In this case, we were interested in spindle scaling, so how the spindle becomes smaller and smaller as the cells are becoming smaller as they are dividing. And if we jump more to sort of trying to image the whole organism at a later time point, where the embryo is more in the range of many hundreds of microns, so 800, 900 microns, then we collect data like this, which may look relatively trivial, but in fact requires about 8 different views to be registered and stitched to record the entire early morphogenesis of these zebrafish embryos. So in this case, all the nuclei are labeled, and in principle, one could follow all the cells here to see how the embryo forms. And the other aspect that I want to highlight here with this video is that, as I said, it's not necessarily trivial to record this data and to reconstruct the embryo, but it becomes even more difficult if you're trying to track all these cells, and especially if the aim ultimately is to not just doing this for a single embryo, but doing this for many embryos. And of course, if you want to do developmental biology on these data sets, a single data set will never be enough. But instead, you will have to deal with, I don't know, hundreds or thousands of data sets like this before you can actually draw any conclusions on how a fish typically develops. And so a lot of the efforts that we've made in the past years and still ongoing are to find clever ways on how to collect this data, ideally in a more in a smart and very efficient way, and also in a way where the data can be compressed, or let's say where the information from the data set can be extracted very efficiently. And I'll give you an example on that a little later. Now, of course, if we go to much larger scale, so if the fish is several weeks old, we get into a regime where we cannot image the fish alive anymore, but instead we have to fix and clear the fish to be able to get any reasonable penetration depth. And I've mentioned light sheet is also ideal for that. But of course, we've now made a jump from the in vivo live imaging to the fixed and clear tissue imaging. And that, of course, is quite a jump. And of course, we are trying to push this boundary further and further, trying to image longer and longer, or at least have the microscopes that give us still reasonable view into the embryo without having the need to stop the development and kill the fish to then get the necessary optical penetration. And so, of course, there's several attempts made with adaptive optics or moving into the infrared, etc., to facilitate that. And that's something we are thinking about quite a lot as well. In the end, obviously, one of the goals could be to merge information from multiple modalities, whether it's live and fixed, or just simply different microscopy modalities, where each gives a certain advantage or a certain contrast, certain insight into the specimen, and one can somehow find ways to merge those informations from the different data sets. So another thing I want to highlight when we talk about multi-scale, this is just another example of this, is that oftentimes, especially when you're dealing with a relatively large, for microscopy terms, relatively large object like a zebrafish larvae, that all of what's done is always in the context of the entire living organism, right? So even if you are interested maybe in like how a blood cell moves to a vessel, you're dealing with data that should ideally be collected across the entire specimen. And some things that you observe in one area may very well be relevant for other areas as well. And obviously, if you're looking at blood flow, it might be interesting to monitor the heart, record the blood flow in there, the cardiac output, the heart rate, whatever, because it will have consequences on the blood flow in other parts of the larvae. But looking at this video, which is, it's reconstructed from multiple movies taken at different time points across the entire length of the larvae, it may become clear that it's not necessarily trivial, or it may actually be impossible to collect such data at a given time point at one instance. We simply have a limited pixel rate from our cameras, which means we cannot see the entire field of view, the entire embryo or the entire larvae in 3D for an extended period of time to record these movies. We can only be at a given point in time at a given location. And at that location, we only have a limited resolution. So unless you deploy, many cameras to look at the entire fish in parallel, you essentially have to make a wise choice where you go to image. And that's something that ultimately runs into the problem that we call smart microscopy, which is making a wise choice where to record the data at a given moment in time during development. And that's something that is a big goal for us, to develop these smart microscopy techniques, to have the microscope ideally know where to image when, and that not just in real time based on the present sample, but ideally also using data that was collected in previous experiments. So I want to give you two specific examples from the past years that I think highlight very nicely how important it is to build a custom instrument for a specific application. Both of these examples could not have been done with a commercial microscope because they simply don't allow you to record the data the way we've done it. And that I think is relevant in the context of the Flamingo, that the Flamingo is also an attempt to be able to build a custom system, custom microscope for a specific application. So to highlight this, I want to first focus on the zebrafish heart. And so to give you a bit of context, this is a two-chambered pump, so to speak, that consists of 3 layers. There's the endocardium in green here and the myocardium in red, and there's the cardiac jelly in between these two tissues. It's about 1/4 millimeter in size, and the goal for us was to reconstruct the heart in 3D. And indeed, that's possible. I shouldn't say 3D, it's probably 5D, because you need 2 colors. You want to do it in 3D, and you want to do this obviously in time. Ideally on 2 time scales. One is the short time scale of the beating heart, and then possibly also on a longer time scale, meaning throughout development, being able to not just see how the cells move during a heartbeat, but also how they behave during development. So in order to collect data or to reconstruct data like this, what's necessary is an intermediate step where you collect data as it's shown here on the right, which is a cross-section through the beating heart. And so what we do is we collect movies at different depth throughout the heart. So we're not taking a stack of images, but we're taking a stack of movies. And then these movies are synchronized because in each movie you can identify a heartbeat. And then by synchronizing these heartbeats, they can be overlaid, and you can then reach such a 3D reconstructions as we see on the left. And so this is typically the kind of data that we then analyze in more detail. In this particular case, we were interested in the mechanics of the myocardium and the endocardium, and what role the cardiac jelly, the extracellular matrix in between these 2 layers plays. And so data like this obviously is instrumental for addressing this question. So again, this was done on a light sheet microscope that was dedicated for this particular application. I would say, the best light sheet microscope in the world for imaging the zebrafish heart, because it also involved not just the microscope itself, but also sample preparation, the way the fluorescence is detected, both colors simultaneously, and then particularly how the data is acquired and how it's reconstructed, something that you couldn't do with an off-the-shelf commercial instrument. So this allows us then to build models on how what happens when the myocardium contracts and what the mechanical consequences are downstream on the endocardium. So the second example I want to give is more on a larger scale, namely zebrafish gastrulation, trying to address several questions here, sort of what is a stereotypic development in zebrafish, where do cells move, when and where, and what does one embryo have in common with the next embryo. And as I said already, this isn't trivial, especially knowing how large these data sets can be. It can easily be several terabytes of data. So as I said, oftentimes we want to rotate the specimen in order to look at it from different sides. Alternatively, instead of rotating it, you can also use multiple objective lenses. And as we've already seen, we sometimes use 2 illumination lenses to illuminate from the left and from the right. And we can also use 2 detection lenses to look at the front and the back simultaneously. And so if you use all of that, so you illuminate from 2 sides and you detect from 2 sides, you end up with 4 data sets. And what's illustrated here is just how we go about collecting this data and what each camera sees. So on the left, you see what the front camera sees. On the right, you see what the back camera sees. And as we are halfway through the specimen, you can see that the back camera image becomes much cleaner than what the front camera sees. So that illustrates how valuable the back camera is. And then if we go back with the stack, you see again the nice quality of the camera in the back. And once again, once we are halfway through the specimen, you see that now the front camera becomes more important and shows you a cleaner image of the specimen. And again, illumination, in this case, it's called from the top, shows you the top half of the specimen in a better contrast than the lower half, and illuminating from the bottom, in contrast, gives you the bottom half much nicer. So each of these data sets basically has a good quadrant of the specimen, and one would want to identify this data and then stitch the data together. Or oftentimes, one can also just cut the data into pieces and merge it that way, especially if the data doesn't need to be registered, which is the case if you have these 4 objective lenses when there's no rotation necessary. Of course, the cheap and simple version of this would be to just use 2 objective lenses, one for illumination, one for detection, and then rotate the specimen. But that can also get a little bit tricky because then the data needs to be registered, and the data merge cannot really easily be done in real time. So with this in mind, we set out to record gastrulation in the zebrafish. And in this case, actually, we felt that 4 data sets are not enough, and instead we are recording 8 data sets. So as you see, the embryo is moved through the light sheet, data is collected from 2 sides, and then the fish is rotated once by 45 degrees, and the procedure is repeated, ending up with 8 data sets, which of course then need to be registered and then merged. If you do this on the full 3D data set, and you're doing this over time, over and over again, you'd be collecting terabytes of data. And with the question in mind, how are we going to be able to do this? If we wanted to do this 100 times with 100 samples, then it would require a lot of not only of hard drive space, but also strategy on how to merge the data and how to compare the data. So instead, what we decided to do is to compress the data in real time. And the idea here is that as the specimen is moved through the light sheet, as is indicated by these little pie pieces, we know pretty precisely which part of the specimen is going to deliver us good data from these individual views. And so we can pick that data and we can generate a radial maximum intensity projection. And so what you see here is not a 3D data set, but it's essentially the entire embryo unwrapped. So in this case, we're looking only at the endoderm. but we see all the cells of the endoderm in this one image. So this image represents like a world map of the embryo. And as I said, instead of a 3D data set, we have a 2D image. And this reduction of the dimensions means that we can heavily compress the data. Of course, we're making certain assumptions here, namely that the fish is a sphere. At this stage, this is a good approximation, and that we can use all these different projections that are available in the geographical sciences, and we can represent the embryo in such a form. And then, of course, if you do a time lapse, and here we're using a slightly different projection, you can watch each and every cell move around the embryo by just looking at this 2D movie. There's no need to use a sophisticated 3D data viewer. You can see everything that should be of interest in this one view. And of course, which is very important, it allows you to look at the data immediately after the experiment. And that is typically not the case, and oftentimes is a big issue that people collect terabytes and terabytes of data without actually being able to inspect the data very quickly. And so here we have the possibility to take such a time lapse overnight, and then the next morning you can look at a little QuickTime movie basically on your cell phone and check the data, and then hopefully make some smart choices of what next to do, which experiment to do next. Also, if you want to visualize the cell movements here, this becomes a lot easier if you're just doing it in 2D, of course, acknowledging sort of this weird geometry of your projection, but it simplifies things quite a bit. Of course, if we don't just look at the endoderm, but look at multiple germ layers, shown here in 3 different colors, then things get a bit more complicated. And so we also have to find a solution for this, where a simple projection, simple radial projection wouldn't really do this justice. And one way of doing this is just visualizing it in 3 different colors. But at the same time, the problem becomes that you want to somehow be able to identify what are the main principle, what is the blueprint of how the embryo develops, and how does that compare between one embryo and the next, right? And just looking at this data doesn't really help very much, because the next embryo is going to look very different. It's doing the same thing more or less, but its orientation will be slightly different, and cells are just in different positions. But the end result, it's still a fish. It's still a fish embryo. And so we'd like to understand how we can compare these different fish. And one way to do this is to use the software that Nico Scherf and co-workers developed, which tries to bundle the tracks that you record. And so it's basically also acknowledging the fact that the tracking will never be perfect. And it's something that we simply have to accept. The data is never going to be perfectly clear, and tracking algorithms sometimes fail. And so with that in mind, we have to give up the dream of tracking every single cell perfectly accurately, but instead deal with the fact that we have short tracklets that we try to understand and try to combine in some way. And so the software allows us to cluster these short pieces of tracks and ideally visualize then how which path these cells take typically, and rather than focusing on each and every cell, because that is going to be different in the next embryo anyway, and instead rather look at sort of the global movement of these cells and tissues, and ending up with some sort of in this case a projection again, which I see as like the fingerprint of the embryo, which basically summarizes the main movements that we've seen in this embryo, and maybe this is a good way to then compare different embryos. So again, I think the take-home message here is that again, to address this question of gastrulation, it requires a custom-built microscope, custom software to acquire the data with these projections, and or software that allows you to post-process the data in a meaningful way to then generate images like this. Okay, so with this in mind, I'd like to move on to the second part. And so this is about the sustainability of microscope development in an optics lab. So this was triggered when I decided to move from Dresden to Madison. And looking around the lab, I noticed that we had gone into various different directions, using light sheet microscopy oftentimes, or most of the time, but using it in various different ways. As I said, always custom-built instruments. And all of these 8 light sheet microscopes were unique in a way, particularly since they were built by different people in the lab using different pieces of hardware and different software. And that made it basically impossible to disassemble these microscopes, move them to a new place, and assemble them again, because the people that had built these instruments were not available anymore, and the software wasn't necessarily easy to use for anybody else in the lab. So looking at these instruments in more detail, we realized that oftentimes the modifications had been rather minor. So for example, in this paper where we did SPIM and OPT, optical projection tomography, in the same instrument, which is incredibly helpful because now you're not only seeing the fluorescence, but also you have a transmission contrast. This really was down to a piece in the software that allowed us to collect the data. Otherwise, it just required an LED opposite to the detection lens to give you good transmission data. But the thing here was all in the software, how to acquire this projection data and then have that registered with the fluorescence data. Another example was magnetic manipulation, which was another dedicated setup in the lab. And here it was again a standard light sheet setup. The only unique thing about this was that there were electromagnets around the specimen. And we did so because we were able to inject magnetic beads into the sample preparation, which was either the agarose sphere around the sample, or we managed to directly inject the magnetic beads into the yolk of these fish embryos. And this little demo illustrates that this allows you now to rotate the fish in the chorion. So even after sample embedding, you could change the orientation of the specimen in a very precise way in real time if you have electromagnets instead of this permanent magnet as shown here. So essentially, we realized that it would be wonderful if instead of building microscopes like this, meaning on big optical tables using all sorts of hundreds of different parts, if we could build something in a more modular fashion. And we had some experience with building small or building simple with the OpenSPIM and also with the EduSPIM, which was a microscope for a museum. But we realized that all of these systems had actually been always a simplification. The idea was always to build something simple, cheap, small, and that wasn't really what we were looking for. Instead, we were looking for a high-end microscope, just as good as the ones that we've been using in the lab, just built in a different way. And so looking at all the systems, we realized that they really have some very common features, which is always an illumination arm, always a detection arm, sometimes 2 or 3 different arms. But we wanted to unify the system and be able to build different geometrical configurations of these elements using the same components. And particularly using the same software also to run all of these. And so when I arrived in Madison, the idea was born to build a modular microscope. And I should emphasize that the goal here was not to build the smallest microscope or the cheapest microscope or a microscope that was portable or any of that. The goal was to build the best light sheet microscope for our purposes that is as good as what we had before, but doing it in a more sustainable way, using the same components over and over again for all the different configurations. And I was happy to have Rory, Todd, and Joe in the team that could handle optics, the hardware, and the software components of this project. And what came out of this was what is now known as the Flamingo, which happens to be actually a rather compact and small microscope. But again, that wasn't really our intention, which is not much bigger than a dissecting stereoscope that you would find in a biology lab. And in fact, you could have this microscope even sitting on a bench like it's shown here, and don't necessarily need a fancy optical table to run this instrument. We also thought a lot about how to miniaturize the illumination path. And the goal was to have double-sided illumination and also have the ability to do the M-spin, which is the pivoting of the light sheet to reduce the stripes that you often see in light sheet microscopy. And we came up with a solution that allowed us to use one galvo to do the M-spin and the double-sided illumination at the same time, using just a few elements like a galvo, a scan lens, and a knife edge mirror. And so this is what is in the core of the microscope, and that allows us to do this double-sided illumination with M-spin. So again, the goal was to be able to build these different configurations that now we call L-spin, T-spin, X-spin, and V-spin for the different number of lenses and geometrical orientations. And indeed, with one set of Flamingo components, you can actually build all these different variants, and you can also go back and forth between the different configurations as needed. So here you can see how these instruments typically look like as they are being built. So we have a T-spin on the left and a V-spin on the right. And the other thing you see is that in the back, we have two of these electronics boxes, and they basically contain everything else than the optics and the mechanics to hold the optics. So all the lasers, there's a mini computer, there's all the controllers, and you'll see all this in the second part when I show you the Flamingo in the lab. And the only thing I want to highlight with this, which is a small walk through the lab in Madison, is that given the fact that the footprint of these instruments is so small, you can actually have multiple different configurations in your lab. And essentially you can choose and pick which one you want to use at any given time. So if one morning you decide you want to use this microscope because you want to run this experiment, or there's someone with these samples, you can then connect the electronics box to that particular device, which is essentially just a mechanical framework of the microscope. And you don't necessarily need to have all those expensive components, namely the lasers, the camera, and the stages present at every microscope. So here we have a T-spin with two 10x illumination lenses and a 16x detection lens. There's the 3D printed chamber. The only thing missing here are the stages that would need to be added to have this system operational. And then next to that is a V-spin with a 25x detection lens and another T-spin on the right. Okay, so coming to the last part of my talk, which is specifically about how we use Flamingo these days. So when the pandemic hit, we realized that it's actually really wonderful to have such a compact instrument. In fact, you could take an instrument home. So I had one of these sitting at my desk in my bedroom back in Madison, giving me the opportunity to work on the Flamingo, even though I didn't have the proper specimens, but at least I could work on the software and documentation of the system. And the other thing we realized that they can also be remotely controlled, and I'll come to that mainly in the second part of the presentation. So this made us realize that this is really very much in line of what we've always been thinking about, namely that the microscopes that we tend to develop in the lab should ideally go straight into the hands of the biologists, meaning that those are the ones that can judge how good a microscope is, and those are the ones that also can give us feedback what needs to be changed in a particular microscope design. And so the idea was born to use the Flamingo in an iterative way, passing it back and forth between the developers and the users, and allow them to optimize the instrument for a given application. And the fact that the microscope is portable is obviously a key ingredient to this, so that the biologists don't have to travel to my lab, where the microscope may be bolted down on a big optical table, but instead the microscope can travel to the biologists. And that means that they can also use the instrument in their normal environment, basically where they have all the equipment and where the samples are at home, where the incubator is not too far. So we can package these microscopes in Pelican cases. We can transport them ourselves in a car, or we can ship these quite nicely. And so they've been to various different places. For example, the Marine Biology Lab in Woods Hole, where we've used them for several courses, but also for some scientific experiments. There's currently one in San Diego. There's one at the MIT in Boston. There are several around Madison with collaborators and co-workers, and we have 3 Flamingos now here in Göttingen in Germany. So again, just to highlight this, this part, that we really think that it's the best if the specimen is where the sample, if the microscope is where the sample is, and it could even be, running in a fish facility or in a virology lab or wherever, so that the samples don't have to travel too far to the next facility. So again, the idea is that the biologists have an idea of what they want to do, and they don't have to apply for funding. They don't have to acquire a microscope, but they talk to us. And this could, of course, be done remotely. We design an instrument that we think is most suited for this application. We can send this instrument to the remote lab, have the lab evaluate it, give us feedback, and we could modify the instrument. And ultimately, when they are done with the experiment, they could pass it back to us or pass it on to the next lab that may need the same configuration or a different configuration, depending on their needs. And then eventually, of course, the system will have to come back. We consider the Flamingo as being a living microscope, so to speak. So there is never just one Flamingo, but there's a family of flamingos. Each and every one will be a different configuration. And of course, they will also evolve with time as maybe new developments come up, or if the needs of people change. We would want to reuse the components as the system matures and as it's being, changing its applications. So just, a few colorful images from different labs, different applications, different specimens. All of these have been recorded on the Flamingo, and oftentimes it has required us to modify the system quite significantly. Most of this was really around the chamber, the sample chamber, that's typically 3D printed. And oftentimes it's about the environmental conditions to keep the sample happy or to allow for certain experiments. So I can probably skip this part and save this for later. But this is about the control scheme of how we control the microscope. I think the demo will probably be most useful for that. But just as a teaser, we can remotely control these flamingos. The data can be streamed to different storage devices, and that allows us to also collaborate very effectively with different labs. And as I said, we think that this is the ideal platform for developing more smart algorithms to move towards the smart microscopy idea. So in summary, I think I've convinced you that light sheet microscopy not only offers high speed and low phototoxicity, but is actually a system that can be tailored to a specific application. And that's why I see a lot of the power also of the Flamingo. You often need custom software for data acquisition and analysis. We think it's very important that the data, meaning also the microscope, is reproducible. It's important that we can bring the microscope to the biologists. The microscope should be where the sample is, and a lot of the other things, meaning the actual image acquisition or the analysis, could be done remotely by experts. But of course, the microscope needs to be where the sample is, but everything else could be done remotely. And we believe that all of this is in line with open science concepts, in particular, trying to democratize the access to high-end microscopy. So with this, I just want to give some impressions from the lab, things I can't show you at the demo, but we have a few 3D printers. We're getting beautiful parts from the machine shop here, and all of these things allow us to build the flamingos. So with this, I'd like to acknowledge people in the lab, current and former members, particularly people from the lab in Madison, and also people that were directly involved with the development of the Flamingo, and acknowledge also support by some of these companies that have given us some significant discounts as we were building all of these flamingos back in the US and also now here in Germany. So with this, I'd like to end, and I'd be happy to take any questions that are related to maybe more the first part or light sheet in general, and maybe then the Flamingo questions we could save for the second part. Thank you.
Thank you very much, Jan, for this wonderful overview of light sheet. So there is one question it's about temperature control in Flamingo, but maybe we can answer that question in the second part, or would you like to answer this now?
Yeah, I can talk about it now a little bit and then show temperature control on the Flamingo. As I said, a lot of the add-ons, a lot of the what needs to be done with the Flamingo now in order to tailor it to different applications are those different add-ons like temperature control. But what's the question about the temperature control?
They would like to know how you control temperature in Flamingo. For example, okay, when you have several fish.
Yeah, I can show that at the instrument. There's various ways. One is we have a perfusion setup where you can pump preheated medium through the chamber, or you could heat the chamber itself, which is something that we have implemented as well, where you have a heating foil that's installed under the chamber, and that heats the chamber and the medium directly. But again, it's those kind of customizations that oftentimes really are the key to a successful experiment, and a lot evolves around how the medium is prepared and how the chamber is heated, etc. Those kind of things.
Sorry. Gregory has his hand on. He has a question.
Yeah, I'm just curious about like how many, do you have problems moving these across from one country to another in Europe? If you loan one, or do they stay in the same country? Or obviously here Alenka and I and people, so I'm in Brazil, and so Alenka in Chile, and then the people in Uruguay. And I'm just curious of, do you run into problems transporting equipment between countries?
Well, obviously, we have to deal with customs and whatever the regulations are within the EU. We haven't done it yet, but we will be doing it very soon. Of course, we are hopefully facing less of those problems, and it should be pretty easy to ship a Flamingo from one country in Europe to another country. Ultimately, I think it would always be the best if this is sort of a long-term project, long-term collaboration, if the microscopes could be built in the respective countries. So I think in the long run, our goal would be to have collaborators in, the main, countries where there's interest in light sheet microscopy, and then instruct these people which parts to buy and train them how to build the instruments, because it not only serves the purpose of distributing the instruments, but also training people, and maybe have the physicists or engineers also learn more about how to build a microscope, and not just train the biologists how to use one, right?
Great. I just will finish here. I'm going to have to go off to another meeting, but I love your images of the heart development. So that's where my real heart is, pun intended. So,
Yeah.
But I think I'm going to quit being the director here and come work in your lab as a postdoc. So, look, I'm sorry, but I have
to come here. And yeah, it's that's the part I must say I enjoy very, very much. Going to talking to the various different people here, to the machine shop, to the electronics workshop, look at the parts coming off the machines, coming off the 3D printers, and discussing, how these need to be modified, and seeing how everything comes together. We have a very nicely illustrated, detailed manual on how to build a Flamingo. We have all the CAD drawings for all the different parts, and we have 3D models of, as you've seen, of all the microscopes. So that's really a lot of fun, tinkering with these parts and putting these microscopes together.
Yeah, just along those same lines of that sort of thing, do you have a protocol manual that you're building so that people don't need to try to reinvent the wheel? So if you have something that you done with zebrafish, it has very well written protocol to do that, or is that online somewhere?
Yeah, some of that is online in terms of like sample preparation, like how to embed the fish in the tubes. There's videos on that, like Jove videos, and some of that I think is on our website, or is going to be on our website very soon. Yeah, we are making an attempt to make all this publicly available. The microscope itself is not yet open source in that sense, because we see it as an instrument that is more like open access or open hardware, in a way that people can apply to get such an instrument in collaboration with us. We don't necessarily want to invite people to, we don't want to say like, oh, here are the drawings, go ahead, build your own. Of course, some people would be capable and certainly interested of doing that, but at the same time, this is more geared towards the biologists, and they don't necessarily have the tools, the workshops, laser safety protocols, etc., in place to build something like this. And that's the main audience for us. And so, I think it's, at this stage, I think probably pointless of just putting all those drawings out there, because there's going to be very few people without lots of training from our side to build such a rather complex system then.
Okay, I just have to, I'm sorry, I just want to ask one more question. That you mentioned something about having, you can also do this with a single objective.
Mhm.
And so, but then, so then you have to have a special objective to do the light sheet that passes through, or is that something comparable to the old Zeiss 5 Live system? I don't know, so if you're
Yeah, that's a
familiar.
good question. Yeah, I mean the 5 Live is like a line scanner, and you could in principle, you know, use something like this to generate a light sheet. So it's somewhat comparable, but the idea there is that you use a high NA lens. So you need first of all, you start with a very high NA lens that allows you to use the same lens for illumination as well as detection. And then these typically don't end up being perpendicular. So you're dealing with the problem that these are less, the angle between these two is lower than 90 degrees.
Mhm.
And so you don't have necessarily the perfect view that you would get immediately. So back in the optics, then you have to use two more objective lenses to actually re-image what you are illuminating with your tilted light sheet, so that ultimately you get a sharp image on your camera. And then you can use galvos to scan this around and, you know, have this always nicely image onto your camera. So basically the deal is you're not using two lenses by the sample, you're just using one lens, but then you have two additional lenses in your detection path. So instead of two lenses, you're using three. And you know, you have to do all sorts of tricks and tricky alignment. So it doesn't necessarily make your life easier. I would say for something big like the fish embryos, it makes things more difficult. So for us, it's easier using just those low NA lenses around the specimen.
Mhm. Okay, thanks very much. Excellent.
Sure. See, there's a raised hand by Tobias.
Yes.
yeah.
I'm sorry. I'm sorry. Just shortly, I think that we can ask. We can discuss this question, and I saved all the other questions that are in the chat. So just for honoring the time, that we can answer this one, and the rest we will do in the second part. Okay. Thank
Okay.
you.
Thanks, Pamela. Thanks, Jan, for a really interesting talk and very fascinating. Thank you for the good overview and illustrations of the different types. Very good introduction, I think. So I just want to follow up. So from the LibreHub perspective, from our sort of average program for open source hardware in Latin America for bioimaging, I have these two perspectives that I wanted to ask you about, maybe to follow up on. So you already briefly mentioned open source hardware. So that would be one part of the question, maybe a little bit more in detail. So for something to be open hardware, you have to release it with the proper licenses so that it's open for modification and also commercial reuse. So that's one part. Open hardware can be really expensive or really cheap, so there's a big spectrum. But then in particular for our project, we're interested in things that aren't so expensive. So in using appropriate components, basically, that are more available around the world and maybe not so expensive, so that scientists around the world can actually replicate these kind of systems, basically build them themselves, or maybe have access to kits or share, maybe get hold of a basic set, but then be able to sort of modify it and develop it themselves, I guess. And so I realized that these are two questions. So maybe you can expand a little bit more, for example, on the sharing, whether you already use these kind of open hardware licenses, even though you're not sharing maybe all the necessary files yet. And then on the cost part, where you see there is most potential maybe on sort of reducing this a little bit without necessarily giving into quality, right? So the idea is still to have a research grade item, but for example, by the use of industrial cameras instead of scientific suppliers or something like that.
Yeah, yeah, I think you raised a lot of interesting points there. I think for us, we wanted something quickly. We wanted a high-end instrument that would use the same components that we've been using before. So basically building on our experience with certain hardware components and building on the existing software that we already had. So we basically didn't look very far. We used the same stages that we've been using for the past 10 years. We've been using, scientific grade CMOS cameras. We've been using the same lasers throughout all these projects. So we looked around a little bit, but we didn't try to go very far, trying to, you know, find the cheapest component or, you know, whatever. So of course one could do that again, like one could build a cheap Flamingo by specifically looking for components that are more readily available, that may offer similar performance, maybe not quite the same performance, maybe a bit lower performance at a competitive price. And that could certainly be done. It's just something that we didn't dare to do because it seemed like a high risk step in our situation. I guess once I'm more established here, and let's say we have a couple flamingos, like 5, 6 flamingos running here, you know, we could make this a student project to, you know, to slim one down and build something simpler and maybe like 3D print the whole microscope instead of having, you know, heavy expensive parts from the machine shop. So that's maybe, yeah, part of the answer. The other answer is that, as I said earlier, I think we specifically wanted to tailor this for people who are not able to build a microscope, because our experience with the OpenSPIM, which is essentially an open source light sheet microscope, has simply been that there's been very, very few people that adopted the system, and those have all been physicists. They use this as a starting point to build their own light sheet. Some of that never flew back to us in a way that we got feedback from these people. They just took it and went away, and it never really benefited the people that were really in need of light sheet microscopy, namely the biologists. And they were, some of them recruited a physicist to build a microscope for them. But with the Flamingo, the goal was always to have a turnkey system ready for someone who wants one, right? And in terms of open hardware, it's not something that we've decided yet precisely how we want to deal with this. There's no particular license model yet. But we'll have to come up with one very soon, I think. And what I meant, I guess, with open hardware was basically if somebody explicitly wants to collaborate with us, they are more than welcome, and we're happy to share everything we have. Or if anybody has a specific question, we'll happily share our experiences and our drawings. But we are not yet really ready to throw it out there and have anyone sort of take it and move along and, you know, develop their own branch of Flamingo, because I think it is pretty involved in like, we can build a Flamingo in a day and maybe another day for the electronics box, you know, but it's not something that anybody else can easily do. And I think I feel at least that in order to make it open source, it would probably take much more in terms of documentation and maybe simplification of the system. I would think a lot of people would look at it and say, like, why the hell did you do it this way? And some of that may just have historical reasons of, you know, because it's how we've always been building microscopes or so. So anyway, that's my short answer to, you know, an interesting topic that we can probably talk for hours about how to go about doing this.
Thanks. That makes it clearer, definitely, from the perspective. You know, there's always a certain application goal and where someone is coming from. That makes a lot of sense. Thanks.
Sure. Thanks.
All right. Thank you all for your questions. As I said, I saved them all that were in the chat. I was thinking maybe 5-minute break, coffee break, so that Jan can to the lab, and then we can continue. Thank you.
Okay, wonderful. See you there.
If you still have some questions, you can write them in the chat. As I said, I will save them all for at the end. And if we don't have enough time, then we will be sure to answer the questions and send you the answers. Okay. So now we will start with the second part of this webinar, where we will see Flamingo in action.
Yes. So Can you hear me and see me? Okay.
Yeah.
Just good. So this is totally improvised, just as a warning, which also should allow you to ask any questions you may have. And I really would ideally have this as discussion based. So I want to see what you are interested in, and then hopefully I can show it to you. But just as a brief introduction, so we have one Flamingo here. This is a T-SPIM configuration, so it has 2 illumination lenses over here and here, and the detection arm is hidden right now in the back. And then this on the side is the electronics box. And so I'm just going to walk you through some components here, and then we can go into the demo part, or we can first answer some questions, and then I can give you a quick demo. So let's start with the electronics box. So this is a home built frame, which holds the main element is the laser engine. So the big box over here houses 4 laser lines, 405, 488, 561, 640. And the laser beam from these lasers is sent to the microscope via an optical fiber. And the other things in this box are a mini computer. So there's an Intel NUC, which is this small computer down here, which acts as the brain of the Flamingo. It's not any, sophisticated computer. It's not meant for data analysis. It's just to run an experiment. There's also the stage controllers for the translational stages and the rotational stage. There's power supplies to power some parts here in the box, as well as parts on the microscope. We have a switch here. We can connect different network devices if needed for storage, for example. And then there's also, you may see the antennas back there. There's also a Wi-Fi router. So if you're in the middle of nowhere, you want to connect to the machine, you do it via a hotspot that the Flamingo generates. So speaking of control, this electronics box has a small computer and a monitor. The monitor in this case is a little bit bigger than what we usually have. Normally there's a small monitor just attached to the box here. If we ship the instrument anywhere, it would come with a small monitor, but you can attach your own monitor if you want to. I briefly skipped over the control. I'll demonstrate the control, but you can already see here there's no mouse or keyboard attached to the computer. It's not like a regular, let's say, confocal microscope, which comes with a big Windows computer. In this case, there's Linux running on this mini PC, and you have to bring your own control device, which in our case is a MacBook. And we are connected to our network here, but you can also wirelessly connect to the Flamingo and start an experiment. I'll come to that in the demo part. So this is basically all there is in the electronics box, and everything else is now on the microscope. So the laser light is guided through the optical fiber to the microscope. It ends up coming in here. There's a collimator and a beam shaper here that expands the beam to the proper diameter. There's a slit aperture with which we can tune the beam a little bit. And then it's sent via a few mirrors here into this black box here. This is where the galvo scanner sits, that directs the beam onto a knife edge mirror, which sits on the back side of this cube. And that directs the beam either into the left or the right illumination arm. The galvo also acts as the pivoting mirror to pivot the light sheet to reduce the stripes. And it can very, very quickly switch between sides. But what we can't do is double-sided illumination, simultaneous illumination. We can only illuminate from one side or the other side. And that way have the ability to extend our penetration depth of the light sheet. The chamber is sitting here in the center of the objective lenses. This is a 3D printed chamber similar to this one. So it's a transparent material that we use, which is biocompatible. And you can see it's rather intricate geometry, which allows us to also do perfusion. So there's hookups for tubing, which you also see here. So you can do perfusion or simply use the tube to fill the chamber with medium. There's also an overflow, so you can have like a constant medium refill, and then the overflow will just go to the waste, or you can recirculate it if you want to. These chambers are made for, in this particular case, for a 10x or 16x Nikon detection lens. So you see there's ports for illumination and detection. One of them, this one is a little bit bigger, that's for the 16x. So typically in a T-SPIM, you either put it in one way to use the 10x, and then the other port will have a plug, which you see over here. Or if you decide to use the 16x, you just turn it around and use the 16x port. And there's rubber rings here to seal the lens against the chamber. This chamber also has a heating foil attached on the bottom, and there's a little temperature controller here. So that makes sure that the temperature is set to a constant temperature here. And there's a temperature sensor here that measures the temperature in the chamber. The plug also has a little LED installed inside the plug that gives us transmission contrast. And then on top here, we have the stages X, Y, and Z, and then a rotational stage with which we can turn the specimen. This system has a relatively long foot. We can install different feet of different length. This one is relatively long because it gives us the ability to also change the tube length. So there's a choice of 2 or 3 different tube lenses, which allows us to change the magnification. So if you have like a 16x lens, for example, you can extend the tube lens and get an extra 2x magnification, and that way turn the 16x into a 32x, which makes perfect sense with the high NA that this 16x lens has. Typically, we use PCO cameras. You can't see it right now, but there's a PCO Panda attached here in the back. In fact, I can take off the camera. Go around here. So here you go. There's the camera sitting in the back. This one can also be upgraded with a PCO Edge if you want higher frame rate, cooled CMOS chip. Then that's the next upgrade from this camera. But the Panda works extremely well for our purposes. And so you can see the extent of the detection arm, which also has the filter wheel here in the center. This part here, which is also our own design. And then that's the tube lens back there and the camera on the bottom. So all of this is very modular. The stages can be changed. The camera can easily be changed. The tube lens can be changed. You could in principle use any other laser engine if you wanted. This one works quite well for us. It's the Toptica CLE. If you need higher laser powers, we can also use the Emily variant, which offers more lasers and more power if needed. So that's the quick walkthrough through the microscope. The next thing would then be to talk about samples and sample preparation and how the sample is inserted into the microscope. And I can then also talk more about the software. Do you have any questions about the mechanics of the device so far?
I do have here a couple of questions. Well, the first one is, why is the system called Flamingo?
Yeah, so to tell you the truth, it was chosen as just being a nice name, which is somewhat unique. I don't think it's necessarily a name that Zeiss or Leica would choose for their microscope, so we just thought it would be a cool name. I guess also now you could argue that this is like a single leg that the flamingo stands on, kind of like a flamingo. We also have some magenta colored parts now that make it look more like a flamingo. So now we can also build an entire flamingo that's pink or magenta. The flamingo is also a migratory bird, so it likes to travel. And so you can come up with different arguments why flamingo might actually be a nice name, but it's, yeah. In the beginning, it was just chosen as being, you know, a pretty name, a cool name that hopefully everybody remembers, that sticks in people's heads. So hopefully tonight you will still remember what that cool microscope was called. So here are some true flamingo colored parts now. So these are magenta anodized from the workshop here. Here, one part that I particularly like, which is the filter wheel housing. So you can see it's, you know, it's not necessarily something that every machine shop can easily make. It's not necessarily something that you would use from a 3D printer. So just to give you an idea of the nice job that the machine shop does here in the physics department. In the past in the US, we've also used services like Protolabs, where you can essentially just take a CAD file, upload it to their website, and they generate a quote within 24 hours. And a couple of days later, you would have these aluminum parts in the mail. They do a terrific job. It's just as good as what we got from the workshop. But of course, if there are small modifications that you may need, the workshop it will be a much better choice. And you know, it's then all about the fit. These parts also go well together. And yeah, and the machine shop also brought up the idea of anodizing these parts. Usually we haven't really anodized these aluminum parts at all. We thought it would be maybe nice to have them clearly anodized, but then they brought up the flamingo colored, and it does actually look really cool, especially if you combine it with some of the black Thor Labs parts and some of the 3D printed parts.
Yes, it does. I'm a witness. There's another question regarding the 3D printed. So it says, how much of the microscope is 3D printed? And what are the parts that are still in metal?
Yeah, I would say probably a lot of the parts, even those custom aluminum parts, could probably be 3D printed. In fact, a lot of these parts, when we designed them, were first 3D printed, just simply to test the fit, and then later were made out of aluminum. I would say something like more critical and structurally important, like these ones, probably still best to have them made from aluminum. So this is this part over here, which up here holds the objective lens. So these would, ideally, I would still want to have these made from aluminum. Again, this is a standard piece. This is the same piece on all three sides. So it's the same piece for illumination arm, detection arm. So we typically need like many of these, in fact, 3 or 4 per microscope. So the machine shop just made like 20 of these for us. And then there's a matching cap that goes on top here to hold the objective lens. So all the parts that are like silvery aluminum, those are from the workshop. So namely this part, this, and then these plates here, the cap here. So all of these, this is a standard Thor Labs part, but all of these come from the workshop. And then 3D printed, for example, here, the adapter to hold the rotational stage. So, you know, these kind of parts, kind of cool with the Flamingo logo here. You can see it. These come out of the 3D printer. And you would think these are like, really important sort of for the integrity of the stages, the microscope, the stability, but these work just fine. It's no problem with these ever. And of course, the chamber is 3D printed, obviously, because this is some geometry that you could not ever make with subtractive manufacturing. So this would need to come out of a 3D printer if you really wanted something so complicated with these different channels and everything. So currently here, 3D printed little things like this, like the temperature control holder, the cup holder here. This plate actually here that holds the stages in the back is 3D printed. This is 3D printed, this adapter here. There's a few 3D printed parts. In fact, we also have a cover here that if you have the room lights on, you want to protect the microscope from any light hitting the detector, the camera. So we like to put this cover on. Slides on here. Nice thing is you can try different fits. You can print it, see if it fits. If it needs any modification, it can be very quickly done. Just print another one. There's also a little cover here that we can put on top of the chamber to make it more temperature stable, avoid evaporation a little bit more. And then I was mentioning the V-spin configuration. Over here, you can actually see a V-spin. And on this one, for example, the entire foot on the bottom is 3D printed. Obviously, it has this cool organic shape that you couldn't really do out of aluminum. But I was told that this is actually also more stable than if you made this out of aluminum.
Speaking of
as I said earlier, you could probably print the entire microscope if you wanted to. I don't know if it would necessarily provide the same stability, but it would definitely be worth a try.
Now that you were talking shortly about the V-spin, there's one question here. For in vivo live imaging of the CNS in rodents, the most appropriate spin variant would be the V-spin?
Yeah, probably. I think there's a lot of, as I said earlier, a lot of the choices you make when it comes to the geometry, it's really about the sample, how it likes to be oriented. I think even if you did neuroscience in zebrafish, we probably wouldn't want the fish to be hanging in the microscope, but rather have it in its natural orientation. And that's something you can do very well with a V-spin. It's designed to look into things from the top. It's not really from the top because you always have this 45-degree angle. We talked about the single lens light sheet designs that could actually be more appropriate for something like this, because then it's really just one lens that you have to get through a little hole in the skull in the case of a mouse. But yeah, I mean, that's the whole point of the Flamingo, that you can take a system like this and convert it into something like a V-spin relatively quickly and simply try out if it is the preferred geometry, and maybe print different feet that allow you to change the angle under which the lenses would look into the specimen.
Okay, there's one question about alignment. Is it user-friendly, and how often do you need to align the system?
Yeah, it's the system itself is extremely robust, surprisingly robust, I must say. Sometimes, we take the instrument from here, from the physics department over to the biology department over like cobblestones, and it doesn't lose alignment. We set it up there. It doesn't need to be realigned. Sometimes when we ship the microscope, of course, we go through the alignment process. It's something We can, of course, do ourselves, or we can instruct people how to do it. It's relatively simple. All those mirrors that you need to touch are open, are accessible. We remove the thumb screws here, so you need an Allen key to turn those screws and move those mirrors and align the microscope. Some of these are clearly labeled. It's clear which ones to touch and which ones not to touch. And I think it's relatively straightforward to align the system. We have a little calibration mirror that you stick into the chamber instead of a sample. And it allows you to see the light sheet on a grid pattern. And on that grid, you can then position the light sheet. You can measure the waste. You can see where the waste is. And that helps tremendously to do the initial alignment. You can go to fluorescent beads if necessary for some other alignment steps. These mirrors, we make them ourselves. There's a photolithography lab in Madison where we made these. We just gave them a PDF with different targets, and you know, we give those away. If you want one, we can send you one. And we put them in FEP tubes and then insert them here and use those for alignment. So in principle, it's something that anybody can do. It's for a few simple steps.
Okay. Regarding the sample size, what are the current limitations of it? So
Yeah,
the sample chamber size, sorry.
Yeah, I'd say there's not really, in terms of light sheet in general, there's no real limitation. Of course, in terms of the Flamingo, limitation is working distance of the lenses. So these water dipping lenses that we typically use have a working distance of about 3 millimeters. And that essentially sets the upper limit of the sample diameter. So typical samples are more in the 1 to 2 millimeter size range. The embedding we do is in mostly, like for a T-SPIM like this, would be in FEP tubes. And these tubes have a diameter of like 1.5 to 2 millimeters or so. And they need to fit between the lenses. So I don't know how well you can see this, but here's a zebrafish in an FEP tube. So this gives you an idea of how small these tubes are. And so there's a fish inside, a fish embryo inside here, and then this goes into the stages, into the dipping into the chamber. These FEP tubes come in various different sizes. We buy them in meters, and yeah, you can buy all sorts of different sizes depending on your specimen, different wall thicknesses, and as long as they fit between the lenses, it's all good. But of course, if your sample was any bigger, let's say bigger than 2 millimeters, in that case, you would probably turn to a different setup, mainly using different lenses. Ultimately, it's all about the working distance of those lenses. You know, we've used the 4X Olympus lens in the past, which allows you to look through air into water. So you can have a glass cuvette that's like 1 centimeter or 1.5 centimeters in diameter, and have those lenses look through the glass window onto your specimen. And there may be other water dipping lenses that have longer working distance than the ones that we use. So there's no real limit, so to speak. It's all about, and as I said, for clear tissues, samples oftentimes are much bigger, and then obviously you'd be using lower magnification, longer working distance lenses.
There is one question from the previous part regarding illumination. So it says, you mentioned infrared illumination to improve penetration. And the question is, are there any negatives to ER? Yeah. Sorry, negatives to what? Are there any negatives to infrared, to using that illumination?
Yeah, of course. I mean, so if you wanted to use infrared, the obvious advantage would be that in theory it gives you better penetration. Longer wavelength means less scattering. The The problems are typically manifold. One is you would need the proper probes, proper dyes, or ideally fluorescent proteins that fluoresce in the infrared. Those are rare or sometimes simply not available yet. The other thing is that if you want to illuminate with infrared, ideally you also want to detect in the infrared, meaning you would need a sensor, camera sensor that's able to detect the fluorescence. So those cameras tend to be very expensive and have relatively low resolution. So that would be difficult. The other thing is that you can't go very far into the infrared because of absorption. So there's only a small window that you would want to use where infrared is really beneficial, because ultimately there's going to be a lot of absorption that you want to avoid just as much as you want to avoid the scattering. So you have to be careful what window to choose. If the question is more about 2-photon, I'd say 2-photon is just a whole different topic. The main benefit in 2-photon microscopy is that you can generate a tiny, tiny little volume of fluorescence. Whereas here, the goal is to illuminate an entire plane. So 2-photon illumination is simply, a poor choice for something like a light sheet. So if the goal is more to increase depth penetration, then you would want to use infrared illumination and infrared detection.
Okay, there is one that came up now. For the translation of the sample, do you have piezo or step motors? And sorry. And on the electronics for controlling the movements and also the lasers is also custom made?
Yeah, that's a good transition maybe into a software demo. We use regular micromotors, so no piezo stages. It could be anything really. I think the rotational stage is piezo driven. Some of these piezo stages can be very, very fast, but they don't necessarily have the long travel range that you sometimes need, especially along the vertical axis if you have multiple specimens lined up on top of each other. But that's not really a limitation. You can choose whatever stage you want for light sheet. We happen to use these PI stages that work quite well for us and are very well compatible with our software. So speaking of software, we have our own software to run the microscope. It's all sitting, well, half of it is sitting on the little Intel NUC. It's running Linux and it's running a software that controls all the hardware components. And the fact is that the control of these different hardware components is relatively easy, because all you have to do is move some stages, trigger the camera, trigger the laser, move the filter wheel, those kind of things. And it's something that, in fact, a Raspberry Pi could do. And we've had Raspberry Pi in here before. But when it comes to high data rates, and especially with the edge, with the PCO edge, like dedicated frame grabber cards, then ultimately you need a PC of some sort. And so that task is relatively simple and can be nicely done on this mini PC. The second part of the software is what's running on your laptop. And so maybe I can show that briefly, what that looks like. But maybe before we get into that, just want to show you again. So the specimen here, in this case, the fish is embedded in a plastic tube. And this is inserted here into the microscope. And it's then going to be placed into the light sheet. I can actually also show you the light sheet briefly. I don't know if you'll be able to see that, but try. So again, here, you can maybe spot the illumination lenses on the left and right, and we see the front of the detection lens there in the back. So this is the 10x detection lens. And if you try to put this on top of the stage here. So if we look through the stages, maybe hard to see, but you can see there's the 3 objective lenses. So the one in the back is the detection lens. Left and right are the illumination lenses, and you can maybe see at least the 488 laser a little bit. They're scattered on the surface of the lenses. So this is where the sample will go. So first, let me switch off the laser. Okay, so again, here's the specimen and the tube. This just gets inserted like this. So let me try to switch cameras. Okay. So briefly say sort of a bit more about the concept of the electronics. So the idea here is that the microscope, the Flamingo itself, does not come with a computer. You have to bring your own computer, which in this case needs to be a Mac. So you bring your own MacBook laptop. You start the Flamingo software, which we see here on the screen. And probably can't quite see this, but here I can choose from some of the Flamingo instruments we have in the lab. And I can then connect to this microscope, and I can see some parameters here of the components. I can then switch over here, design an experiment, set all the settings that are relevant for my experiment, and then start, let's say, a time lapse for the next 24 hours or so. I can then disconnect from the machine and connect to a different microscope, let's say the Huisken over there, and then start an experiment there. And all of this can be done remotely. So I don't even need to be here. I can do this from home or from my office. Of course, somebody needs to be here to put the specimen into the microscope. But other than that, all of the controls, all of the experimental design, so to speak, can be done remotely. In this case, we are not remote. So we are in front of the microscope. So we also have a preview screen here. So as I mentioned earlier, this is a larger screen than usual because we have the space here. If we were to ship the instrument, it would be a lot smaller, or you bring your own monitor. And so I'm not going to walk you through the entire software here. But yeah, you can maybe not. There we go. So you can see that the sample that I have just inserted shows up on the preview screen. So we're just going to take out the sample one more time, then put it back in. So that's sort of the initial view you get. You can actually mechanically also rotate the sample by just rotating the sample holder, or you can rotate the sample through the software. There's all sorts of settings here that I don't necessarily want to go through here unless you have any questions. It's the typical settings you would find on any other fluorescence microscope. So we can, here on the top, we can choose the illumination source. In this case, we've selected the LED. We can select any of the laser lines. We can move the stages either by clicking on buttons or moving these sliders, or what I prefer is using the keyboard. So for those of you who've played computer games, you know some of these keys are for left and right and front and back, or accelerate and brake if you're into car races. And so we use the same keys here to move the sample up and down, left and right, and also to then focus through the specimen. So all of this would then be done in preview mode using the LED as a transmission light. And then once you're done with that, you can switch to fluorescence. So in this case, I selected 488 laser line. So you can see the fluorescence here. Now it's probably a good time to also put the cover on. I showed you briefly. It's a bit more contrast. Obviously, normally you would want to have the room light switched off, so you get a bit less background. So here we see a bit of a room light as a background. And now I can use the stages to move the specimen again back and forth, and also here focusing through the specimen. This is the back side. So we would want to be over here on the front. So we can change illumination sides. Here in this case, we're illuminating from this side, as you may be able to see. Now we're illuminating from this side. So in a typical recording, you would illuminate from both sides, like take a stack with one illumination arm and switch the illumination, record another stack from the other side, and then merge that data. The sample can also be rotated, as I said, in software. So as I've demonstrated in my talk, you may want to take, different views from different angles. So here, this would be a side view. Actually, here you can see the heart beating quite nicely here. So all of this is live. So it's basically streaming with 40 frames per second. So you can see live what's happening in the specimen and make your choices in terms of which plane you want to image, or which part of your stack you want to acquire. So all of this is configured over here. And there's also a time-lapse module where you select how often you want to image for a given time your specimen. And all of that is basically pre-configured, and then it's saved to a workflow file, which is essentially a tiny little text file, which acts as a script for the mini PC to then run as an experiment instruction. So it has all the instructions what the microscope should do, when and where. And that is the file that is generated on the laptop, that is sent to the Flamingo. And then once you click start, the Flamingo will record the data. And that's the moment where you can close your laptop and walk away. At the same time, something that I haven't mentioned yet is that the data is actually saved to wherever you want the data to go, because the Flamingo does not come with any storage. So I forgot to mention that, but you can actually see it here. This is the Intel NUC. And right now, all we've connected here is a little SSD drive. So these are incredibly fast. They come in 1 terabyte, 2 terabyte, or 4 terabyte sizes. So that holds an entire experiment most of the time easily. The benefit of this is that you do your experiment next morning, you just unplug the drive, take it with you to your analysis machine or your laptop, whatever, to look at the data. Of course, in a setting like ours, we haven't actually set it all up yet, but we're in the process of setting that up. We will be streaming the data via the network to a file server. So there's 10G copper here, and there's also fiber to a file server, which is just across the hall here. And so we can stream the data there, and hopefully we can do this in parallel for all the different Flamingos in this room. And the idea behind all this is that, as I said, one person puts the sample into the microscope, another person could be the same, but it could be a different person, starts the experiment, designs the experiment, runs the experiment. It could also be a computer sending this workflow file to this machine. The microscope spits out the data, sends it to a network attached drive or a cloud, and then a third person could analyze the data, modify the workflow file, and run the next experiment. So I like to look at it as this is not a microscope anymore, it's more like a robot, or it's a machine. Input is the workflow file, output is a set of images. You could analyze those images and then redesign your experiment, change the workflow file, get the next set of images. And that, I think, is the ideal way of how a time lapse should be run, so that you're not just predefining the time points at which the data should be acquired, but rather take one data set, analyze it, and then make a wise choice when the next data set should be acquired and which part of the specimen should be imaged next. So maybe another feature that I would want to highlight here is that here in the software, as I said, we have the ability to look at the sample live. So, for example, you can see that it's live because the heart is beating here. So this is a live preview. But of course, the disadvantage of this running in live view is that we are constantly acquiring data. So in this case, the sample is permanently illuminated. And we are streaming 40 frames per second to the computer, and we're exposing the sample continuously. So, within those 20 seconds that I was just talking to you, illuminating this plane, we have taken 800 images. In fact, a single image would have been enough to see where we are. We don't need 800 images. Of course, we wouldn't see the heart beating, but one image would have been enough. And so we've implemented a nice feature that allows us to only acquire an image if somebody has changed. So now with that enabled, you see the heart is not beating anymore because this is a frozen image. And only if I move the stage, the camera takes an image to give me an update where we are in the sample, but then it stops acquiring. So it just gives me, it moves the stage and it gives me one update, one image. And that's perfectly enough to see where I am in the sample, and it's much more gentle on the specimen. So the typical way to then record a stack would be to go to sort of the final plane, select this, and then maybe go to the front of your specimen. Yeah, you find this as your start for the stack. And then the typical things you have to do is to define the Z spacing. So in this case, we say we'll require every 3 microns, for example. And then in this case, we are taking a stack of 725 microns, which ends up being 242 planes. And I can do left and right recording, stack recording. Now you can see the stack is being recorded, so you can see how quick this goes. So This is one stack. And this was one side illumination, and then this is the other side. Nothing is being displayed on the screen because the goal here is to be as fast as possible without any downtime. So that the data is streamed directly to the drive and no attempt is made to display this immediately. However, we can activate a feature that gives us a maximum intensity projection. Now you see the next stack is quiet. And what's displayed on the screen, it's a bit saturated because of the camera, but it looks really pretty on here. Here's a maximum intensity projection. This was first with illumination from the right, which looked really nice over here. Now this is illumination from the left, which isn't terribly useful because it's mainly great. It's great for the head here and the yoke if you're interested in the yoke, but it creates some bad shadows over here. So maybe we'll turn this off. And, do this one more time. I can show you maybe. It's long enough. So that's what it looks like. So, and again, that's just the maximum intensity projection. So this is obviously an entire 3D stack that is just displayed in this maximum intensity projection that's calculated on the fly to give you an overview of what the image looks like, and you can judge whether everything's working well. The raw 3D data is written to the drive, and you can now start your time lapse and watch the embryo develop for the next 24 hours or whatever your plans are. Good. So, any more questions about this part?
Well, I have a couple of general questions, and I have one that is related to the algorithm for showing the 3D data.
Yeah.
So it says, when using the unwrapping algorithm for showing the 3D data, do you know if it will introduce any kind of deformation, like in a world map, if the idea is to study morphology instead of trajectory?
Yeah, I mean, of course, it does deform the data in some ways because it's obviously distorted the way it is, just like a world map is. But there is no perfect way to represent what's happening on the sphere in a 2D image. Fortunately, there's hundreds, literally, of different map projections one can use. So, for example, the Mercator projection looks pretty terrible, has all these distortions, but it preserves angles. So if you're looking at the angles at which cells move towards each other or towards the midline, the Mercator projection is angle preserving. So that would be a good choice if you're interested in the angles at which cells are moving. There's other projections that are area preserving. I don't have an example here, but they typically look like orange peels. You may have seen those maps where it's like several ellipses together, where you have like carefully unwrapped the earth in these orange peels. Those are area preserving. So it's just a matter of which one you use. But interestingly, the way we store the data, we don't make any assumption at that point. We don't make those deformations. We save the data in spherical coordinates. So essentially, for each vortex on the surface of a sphere, which has a particular spherical coordinate address, we save the gray value. And then only later you transform this data in a particular way to visualize it. So the only assumption here is that you assume the embryo to be somewhat spherical. And I know people have done something very similar with cylindrical geometries in Drosophila. The Drosophila embryo is like this cylindrical object. And you can take a fly embryo and unwrap it in a similar way, just using cylindrical coordinates.
Okay, there's one question regarding thermal and mechanical drift. Are the 3D printed material from the stage good enough to avoid it?
I don't think I can really answer the question. We don't have enough experience with temperature drift. Of course, there have been ideas of taking such an instrument and putting it into an incubator. We've actually also put these flamingos in little tents and have heated the tent, which in general is a nice way of temperature controlling the sample. If you don't want to heat just the chamber, you could just heat the entire instrument. Of course, you could heat the entire room, but if that gets awkward, you could heat just the microscope itself. And then we've experienced a lot of drift. So this wasn't ideal. I don't know where the drift came from. I don't think it's necessarily the 3D printed parts. It may be anything in that microscope. So I guess fact is you would then have to realign it, and hopefully it stays stable if you keep the temperature stable.
Okay, I have 3 more questions, and I think that we're done.
Okay, it's good to see that there's still some people around.
The first one, now, how much would this entire microscope cost?
Yeah, great question. I must say we've negotiated some good discounts with some of these companies, because obviously we've bought a lot of stuff from them. So that may influence the price quite a bit. Prices can be anywhere between, let's say, $50,000 to $100,000 or euros, whatever. Really depends on what camera you choose and which lens. Some objective lenses can be very expensive. Stages, most expensive are the stages, the lasers, and the stages, lasers, and camera. And then the objective lenses can be expensive as well. And as we discussed earlier, it's again, the idea was not to build the cheapest microscope ever. Certainly, you could build something very similar for much cheaper if you were to just say 488 nanometer is fine. I don't need 4 lasers. You don't need to motorize all the stages. I mean, at least for what we just did, which I think is, a great example of how easily you can acquire such beautiful data set in just a few seconds, can be done with, a much simpler camera, single axis motorized. The rest could, like the positioning of the sample, could be done manually. So you could, if you don't care about rotation, you don't need to motorize the rotation. So there's a lot of things you could skip and make this microscope a lot cheaper. I guess the good thing is that the Flamingo geometry isn't like the backbone of the microscope isn't particularly expensive. So that's the cheapest part, I would say. So you could start off with basically 80% of the microscope, and then just pick and choose some of the components and find some cheaper alternatives for them, or just downscaling it, saying 1 illumination arm, 1 laser line, 1 camera, 1 motorized axis. The software would be able to handle that, no problem, as long as you stick to the same manufacturers. If you went and used a different camera, then we would need to have the drivers and some time to develop the software.
Okay, there's one question here about commercial availability. So do you see any path for the Flamingo to be commercially available? He says, we're a small startup and open hardware has been instrumental for validating ideas quickly, but when trying to make the jump to a commercially supported system, I couldn't understand how to set such collaboration?
Yeah, that's another great question. Our intent has never been to commercialize the Flamingo. I think there's a special benefit of basically being scientists and able to talk to other scientists about their ideas and their dreams and their experiments and helping them build these microscopes and helping them do the experiments they want to do. And maybe I wasn't clear about our goals, but our goal has never been to build a product, to design and distribute a product. It's always been the idea to help other people and to, tailor these instruments. So I might have said earlier that it's not about, building the same microscope 20 times. It's about having a framework that we can use in the next 10 years, let's say, to build better light sheet microscopes. And so all these flamingos, like you've seen too, and a few in my talk, they are all different. They all look different. They have different purpose. And, I of course, some of the parts could be commercialized and could be a kit of parts. But I feel it's just great having such a, modular framework in a research lab. If someone comes along and says, I have an idea of how to build a better illumination arm, you can start with this instrument tomorrow, take off the illumination arm, build your own illumination arm, but you have a microscope that's working to begin with. And that's not necessarily the case in previous situations like this. Normally, when a postdoc comes and wants to build a new microscope, they typically start from scratch, write their own software and whatever, and that takes forever. So here they have a microscope to begin with. And so that's why I look at this more as a vehicle to advance our science, our microscopy development efforts, rather than, building 100 flamingos and selling them across the world. That's something that other people could do. But the goal is specifically to provide an instrument for free for a limited period of time for a dedicated scientific project. And I think that puts us not into competition to some commercial vendors. Instead, it could be like the entry drug into a light sheet system, and maybe you find which configuration is the best, and then you simply choose that commercial system. And so maybe in the end you end up buying a Luxendo or a Zeiss or a Leica or whatever, but you've tested it all first with a Flamingo. So that's sort of my view today. It may change, but currently I just enjoy working with scientists without any commercial interests. Just fun tinkering with these systems and using them in our own lab. And eventually I'm sure there's going to be 8, 9, 10 flamingos here in this lab and in other labs across campus, and hopefully in many other labs as well. So, yeah.
Okay, just the final question before going to Alenka. What is it was from the previous part. What is the difference between Gaussian and lattice light sheet?
Yeah, that is not a simple question to answer in just a few minutes. I'd say the easy part is to describe what a Gaussian light sheet is. So you're essentially starting with a Gaussian round beam coming off the laser. You scale it, you shape it into an elliptical form, and you use a cylindrical lens to generate a light sheet. And then there's many other optical elements to then eventually bring it into the chamber and illuminate the sample. It's basically a light sheet that's not perfectly uniform, but it is like a homogeneous light sheet sort of across all dimensions, illuminating everything at the same time, more or less, with almost the same intensity if you do it right. The lattice is basically a grid that is formed by some way more advanced optical elements in the optical path, and it's then typically also jittered to generate a more uniform illumination. It supposedly has the advantage of being thinner, and some of these also have sort of a self-reconstructing properties where they penetrate better. But they also come along with more phototoxicity and significantly more complicated optics, which in many cases you have to realign before every single experiment, which is not the case here. It's a lot simpler, but I would argue it's equally powerful for what we are doing. And I think there's also a huge benefit of having a simpler, cheaper, more robust system, because it essentially means you can build 10 of these, you know, maybe for the price of a lattice, at least the commercial lattice. And that way we can run more experiments in parallel and get more throughput and maybe more insights scientifically than with a more complex system that is just more pain to be experimentally used by the users in the lab. So that's the short answer to that.
Okay, Alenka, do you have a question?
Yeah, it kind of follows up on this question because it was very intriguing when you mentioned in your seminar in the first part about a lot of things being able to be imaged in the Gaussian beam that were being done with the lattice light sheet. And I'm really interested in calcium imaging, for example, and imaging in monolayers of cells to utilize the speed of the light sheet. And I would really like to follow up on these experiments using your Flamingo, and I was very interested in which configuration you think would be ideal for just a monolayer of cells. Would it be the V-SPIM, or I know there's like the inverted that you're working on. And I was also curious, how is it easy to adjust the thickness of the optical sheet, or is that something that you can easily do on the Flamingo?
Yeah, so to answer the second question first, it's very easy. I mean, obviously, if it only works across a certain range. We have a slit that we use to adjust the thickness rather quickly. So depending on your detection lens, you would then adjust the illumination. There's different collimators we can use to make, you know, wider beams or more narrow beams. And those could be used to also change the thickness of the sheet. There's also ways to move the sheet back and forth if you want more uniform illumination. It's not something we have on this Flamingo, but on other systems, we are implementing something like this that gives you a thinner, more uniform light sheet. So all of that can be done. Some of that can be done basically as you're looking at the specimen, you can adjust it. Other things require a bit of a remodel, but it's flexible in that sense. The first question, I think it's really all about sort of how much you also want to invest into sample prep and how much the sample preparation is a given and how much you are willing to change that. So, for example, with the V-SPIM, it's obviously easier to use the V-SPIM if you have something grown in a Petri dish or in a cover slip. You can just put it straight into the V-SPIM. Now, is that the best way to do light sheet on that specimen? Yes, maybe, but I'd argue probably not, because you haven't necessarily thought about how to best prepare the sample for a light sheet setup. You're coming maybe from a spinning disk or so, where you have a single lens and you have to have a flat sample sitting horizontally under that lens. Whereas here I could say like, well, you could use this system, you could use that system. It's all up to you. And you may go back to the bench and say like, well, actually, yes, it would actually be cool if those cells were not a monolayer. It would be cool if they were dispersed in a gel, if this was like a 3D cell culture or something. And in that sense, the Flamingo or light sheet in general could inspire you to rethink what kind of sample you've been using for your biology and how you've prepared it in the past. And so oftentimes I'd say, yeah, the VSPIM is a good choice to start with, you know, to be sort of compatible with standard sample preparation. But I wouldn't stop there. I would say like, well, wouldn't it be better if, or can you think of other ways how? And so then taking those sample preparations, looking at those and maybe finding those actually to be more physiological and saying, all right, given that sample preparation, which would then be the best microscope? And so I think if you have the flexibility to use either this geometry or another geometry or any other geometry, like you mentioned, the inverted, which could either be, you know, two lenses like this or a T-SPIM with a lens looking from underneath, all of those are readily doable with the Flamingo. You just take the T-SPIM and put it on the side. You know, there's some details you have to worry about, but in principle, geometrically, it's easy to do. So having that opportunity to use any of these instruments may invite people to think more about how their samples could be best cultured and best imaged. Right. So
Yeah,
it's usually a multi-step process. It's people come with their sample and say like, will it work? And maybe we have a system and we can say like, yeah, we can make it work. But now that you've seen all this, you may want to go back and rethink how you've prepared your sample and how you may want to do it in the future.
Right. Like, do you have experience growing cells in FEP tubing? Have you ever tried?
We have not, but I know it's something that that Ernst Stelzer has done quite a bit, and his co-workers. We have not really gone that way, but I know there's, you know, a lot of people thinking about this and using light sheet microscopy very successfully for it, right? I mean, just imagine you could have an FEP tube full of cells in a matrigel. You can take it out of the incubator, or you incubate it straight on the microscope, or the microscope is in the incubator, or whatever you want to, however you want to do it.
Mm-hmm.
And you can image hundreds of cells in one sample preparation throughout the entire cylinder and time-lapse them and do some analysis on them automatically and pick and choose the ones that you like and time-lapse only those at higher magnification or so. I mean, it is simply a very good way to do, you know, high throughput imaging in some ways. And for example, you can use an X-SPIM, so with 4 objective lenses. You could have one lens being a 10x lens, giving you a nice overview. And the other lens could be a 32x lens. And so using the 10x lens, you get an overview, you automatically classify those cells or organoids or whatever, and then pick the ones that you like the best, that you want to time-lapse, and then use the other lens, the higher magnification, to time-lapse those cells or cell aggregates. So I think, again, sort of coming back to the smart microscopy idea, there's a lot you can do as soon as the samples are readily available in some easily manipulatable way in your sample holder.
Mm-hmm. Thank you.
Could be in a dish, could be in a dish, could be in a tube, you know, whatever. You could have multiple tubes in a dish like this. There's endless ideas, you know, especially with 3D printing and like using molds for the agarose to generate different, you know, landscapes of scaffolds for these cells. And again, it's more a matter of then building the microscope around the specimen to have this sort of, these two elements come together perfectly.
Mm-hmm. Thank you. It's very exciting. I hope we can work on
some nice sample preparation chambers or something to study.
Yeah, and I think it's some of that has less to do with light sheet. It's or sort of the geometry of the light sheet. It's more the sheer speed of the system, right? I think a lot of what I've talked about in terms of, I mean, obviously the high-speed imaging in the heart, but a lot of the other things like the multi-view imaging, for example, that's not something that is specific to light sheet. You can do multi-view on a confocal in principle, just that the confocal is way too slow for the multi-view to make sense, right? But you've seen we can take, beautiful data like this in, I don't know, like 4 seconds or so, right? So what that means is that you can take this data so quickly that you can automatically focus on the sample, you can automatically adjust the brightness, the laser power, you can automatically decide which angle is the best, you can, look at 10 samples automatically and pick the 3 you like the best and whatever, right? It's all just about having a high-speed instrument and then some intelligence to make wise choices what to do next, right? So I think it's ultimately it comes down to the speed of the system that makes a lot of that could make a lot of choices automatically. And I think the Flamingo is a nice framework for doing something like this.
I think so. Thank you very much.
All right. Thank you for such a wonderful 2-part webinar, for the demo, for the talk, for taking all this time to answer all of the questions. I would also like to thank the audience for all of the questions.
Yeah, thanks everyone. Thanks for sticking around so long. I know we went a little bit over time, but I hope this was exciting for everyone to see. So.
It definitely was.
Good.
All right.
All right.
Thank you.
Thank you, everyone.
Have a nice day. Bye-bye.
Thank you. Bye.
Thank you so much.